I am Marta. As Teresa mentioned, it's been 25 days that I belong to the Cybernet network. So I will not talk to you about my project here. I will tell you how my first stay as a postdoctoral researcher happened in Montpellier, France, in the team of neuroreceptors dynamics and function. And I will explain to you, I will try to approach to you all a little bit about the unique mechanisms of mGlu receptor internalization. So all my life as a researcher, I have been interested about GPCRs, G protein coupled receptors, as drug targets. Because GPCRs are 7 transmembrane domain proteins that respond to extracellular stimuli to respond through intracellular changes in the cell, but they are ubiquitously expressed in the human body. That makes them a virtual target for treat all of the diseases that happen in almost all the diseases that happen in the body. And this is why they are the target of about 30 or 40%. Drugs are targeting 30 or 40% of the drugs targeting diseases in the market right now are targeting GPCRs. And among GPCRs, I have been focused in class A GPCRs, specifically in metabotropic glutamate receptors, so mGlu receptors. So mGlu receptors are, as all of the GPCR, 7 transmembrane domain proteins, but they are mandatory dimers. That means that they work in pairs to respond to stimuli. Also, they have another characteristic, and is that they have a huge extracellular domain, as you can see here, formed by the cysteine-rich domain that connects the extracellular domain to the 7 transmembrane domains, and the Venus flytrap domain. The Venus flytrap domain is called like this because it remembers to the plant that has, and it's because it has 2 lobes that accommodate the ligand in the middle of these two lobes, and the ligand, like for example glutamate, that is endogenous ligand, will activate these 2 lobes and then will be closed to reach the active conformation of the receptor. And those are the mGlu receptors. But why they are interesting me mostly? It's because they are brain targets. They are distributed through the brain, all of them except for mGlu6, which is confined to the retina. And we can find the homodimers that we recently, it has been demonstrated that we can find also heterodimers in the brain. So due to its distribution, they are related to disorders such as schizophrenia, Alzheimer's disease, Parkinson, epilepsy, depression, anxiety, and so on. And you can get, guess that if these mGlu receptors are distributed through all the brain, they emerge as targets to modulate these chronic disorders. But nowadays, there is not one drug that is targeting mGlu receptors efficiently to ameliorate brain diseases. And it's because they are not normally very efficient. The adjectives that normally appear for chronic treatments are that they are poorly managed, suboptimally treated, inadequately controlled. And this is a burden because brain health disrupts millions of lives. And it is estimated that more than 15% of health loss is due to brain disorders. And I guess, so okay, so a chronic treatment means a sustained location of the target, because why these drugs, they are efficient by targeting the receptor and modulating it in vitro, but then they are not working in vivo. So one of the hypotheses, it could be like, yes, the chronic treatment will imply that the target has to be there to respond to the drug. And one process that modulates the cellular location of a protein, it's internalization. So GPCR internalization is the process where a GPCR that is located in the plasma membrane goes inside the cell through endosomes. So this movement of the protein will imply that if the drug is in the extracellular compartment, maybe the response will be limited or different if it's inside intracellular compartments. So our question here was, are mGlu receptors internalizing? And if yes, how? To decipher this question, what we did in Montpellier was to use a technology that has been optimized in 2015, which is called DERET, or Diffusion Enhanced Resonance Energy Transfer. So it is based on the long lifetime emission of lanthanides, such as terbium. And the idea is that we are using receptors and we tag them covalently with a fluorophore thanks to a SNAP suicide enzyme labeling. So the receptors are in the plasma membrane, and we are adding to the extracellular medium an acceptor, which in our case will be fluorescein, that will quench the long-time emission of the fluorophore. So this will happen at time 0. However, if the receptor is being internalized, if it's internalized because an agonist is binding to the receptor, or due to time, which is called constitutive internalization, there will be a compartmentalization of the fluorophore versus the acceptor. So fluorescence will be recovered and the light will be emitted. So we will be able to follow the kinetics of internalization of our receptor by measuring the quantity of light released in the cell. With this technology, the question was, are mGlu receptors internalizing? And the answer is yes. All mGlu receptors are constitutively internalized. And what is that supposed to mean? So that means that in the absence of any stimulus, they are going to go inside the cell with time. So for example, here we have mGlu1, mGlu5, that over time they are going inside the cell. We are recovering the emission of light with time. And also for mGlu2, mGlu3, mGlu4, mGlu6, mGlu8, and mGlu7. However, as you can notice in red, when we are adding an agonist, not all the mGlu receptors, not all the mGlu receptor internalization is being separated from the basal one. In this case, there is only agonist-induced internalization for mGlu1 here, for mGlu5, and for mGlu3. And for comparison, for mGlu2, there is not agonist-induced internalization because basal and plus agonist conditions are overposed. We investigated also the internalization profile of heterodimers, specifically of mGlu3 containing heterodimers. In this case, we are co-expressing an mGlu3 receptor to get mGlu3 receptor which is non-tagged, together with an mGlu receptor which could be mGlu2, 4, 7, or 8 that will tag, and then we will follow over time. And with this approach, we will be able to specifically follow the internalization profile of heterodimers. And as you can see here, the only heterodimer that it's been agonist-induced internalization is the mGlu2-3 heterodimer, where mGlu3-4, mGlu3-7, and mGlu3-8, there is no separation between the basal condition and the glutamate condition. So only mGlu1, mGlu5, mGlu3, and mGlu2-3 receptors are agonist-induced internalized. Once we decipher how mGlu receptors are internalizing, we wanted to know how they are internalizing, and we took the dogma with us. So this is the scheme this is the scheme of internalization, the scheme because this is how GPCRs have been thought to be internalized for a long time. I took a picture from a paper of 2006, which is really simple, but it's really illustrating how it is going. So it is, the dogma says that when an agonist is binding to a GPCR, the intracellular compartment of the GPCR is phosphorylated by GRKs. This phosphorylation by GRKs, it increases the affinity of the receptor for beta-arrestin, which will be the adapter to create these clutching-coated pits and induce the agonist-induced internalization of the receptor. So in the canonical internalization of GPCRs, there are two adapters that play a key role. First, the GRKs, and second, the beta-arrestins. And starting by GRKs, there exist 7 GRKs in the human body, but only 4 are ubiquitously expressed and specifically in the brain. But as you can see here in this drawing, even though if they are widely expressed in the brain, they have different levels of expression among microglia, astrocytes, neurons, and even in different areas of the brain. So our idea was to investigate the internalization of mGlu receptors in the absence of GRKs and what happens if we were adding one of the GRKs at a time. And to that end, and for the sake of time, I will only show you mGlu3 among the 8 mGlu receptors that we have investigated, because mGlu3 it's both constitutively and agonist-induced internalized. And what we found was this. So we use the delta GRK knockout, the GRK knockout cell line that is coming from Carsten Hoffman lab from Jena University in Germany, and we investigated the internalization of, as I said, mGlu3. And you can see that in the absence of GRKs, after 60 minutes of internalization, mGlu3 is still constitutively internalized, and it's still agonist-induced internalized here as the red component of the bar. So that means that GRKs are not mandatory for mGlu3 internalization, because mGlu3 can still be constitutively internalized and can still be agonist-induced internalized in the absence of GRKs. However, if we are overexpressing GRK2, we can still boost mGlu3 agonist-induced internalization without altering constitutive internalization. And if the GRK, it's GRK6, the GRK that we are overexpressing in this GRK knockout cell line, what we can see is that in this case, we can boost the two components of internalization, both constitutive and agonist-induced internalization. And we demonstrate that this is phosphorylation dependent because we mutated both GRK2 and GRK6 kinases, and we created the kinase dead version of the protein to investigate if GRKs were acting as pseudoenzymes or they needed the kinase activity to induce this internalization of the receptors. And indeed, we saw that by using the kinase dead version of the kinases, we were recovering the profile got in the absence of the GRKs. So indeed, the phosphorylation, the kinase activity is necessary for the internalization of mGlu receptors, at least mGlu3. And this is for the part of GRKs. And now let's move to beta-arrestins. Arrestins, there are 4 arrestins in the human body. But arrestin 1 and arrestin 4, they are confined to the retina also, which is quite special. But arrestin 2 and 3, those are called beta-arrestins, and they are ubiquitously expressed in the human body also. Beta-arrestin 1, it's arrestin 2. Beta-arrestin 2 is arrestin 3, but we all know them as beta-arrestins. So this is a work done by an ex-colleague of mine in the laboratory of Montpellier. He has studied the internalization of 59 GPCRs, And he found these nice graphs where he found that there are several GPCRs that they can be internalized even in the absence of beta-arrestins. And here he was challenging the dogma already. So we wanted to know, okay, let's take again mGlu3 and check what is going on with the mGlu receptors and beta-arrestins. And here we were surprised because here we used the beta-arrestin knockout cell line that is coming from Asuka Inoue lab in Wuhan, China. And we evaluated the internalization profile again of mGlu3. And as you can see here, when we are removing beta-arrestins from the cells, we dramatically drop the constitutive internalization of mGlu3 without altering the component of agonist-induced internalization of mGlu3. If we could rescue this internalization of mGlu3, this constitutive internalization of mGlu3, without affecting agonist-induced internalization of mGlu3 by overexpressing beta-arrestin 2 in these beta-arrestin knockout cells. So we could see that we can say that beta-arrestins are required only for mGlu3 constitutive internalization, which is unique and not described for any other GPCRs just until nowadays, at least that I know. And we wanted to know, like, okay, so this is weird. So we wanted to know if mGlu receptors could recruit beta-arrestins or not. And to that end, we label our receptors with the Venus fluorescent protein in the C-tail domain. And we label the beta-arrestin with our look to measure breadth recruitment, If the receptor is in close proximity with beta-arrestin, there will be a transfer of energy between the airlook and the Venus, and we will see light. So for the canonical receptors, like delta opioid receptor, which is a class A GPCR that it's internalizing as I said before from the paper of 2006. The receptor is recruiting beta-arrestin, so the breadth signal is increasing when we are adding here, as in the black arrow, when we are adding an agonist. And you can see here a nice increase of beta-arrestin recruitment. However, here in blue, you can see mGlu3. So mGlu3 remains high from the very beginning. And when we are adding an agonist, there is not an increase of beta-arrestin 2 recruitment. And that means that maybe what is happening with mGlu3 is that it's constitutively interacting with beta-arrestins, and that will explain why it plays a role in constitutive internalization. And this was nice to know because this was nice to see that in 2025, in February, I think it was, the structure of mGlu3 has been released in the presence of beta-arrestin 1 in this case. And even if they are adding glutamate to the medium, we can see that mGlu3 is a dimer as expected. It has 2 beta-arrestins linked to it, but it stays in the inactive conformation because normally when mGlus are activated, the cysteine-rich domains, they are overlapping together, and this is not the case. So it stays in an inactive conformation or an inactive-like conformation. So that means that maybe this is what is happening, that there is kind of an interaction, a constitutive interaction of mGlu3 and beta-arrestin. So it started as a quick study about how mGlu receptors were internalizing and trying to find the dogma for mGlu receptor internalization. It ended up in this complicated scheme that was a nightmare to do because we found that every mGlu receptor seems to be a little bit different from each other. And beta-arrestins, for example, here, they are required for almost every receptor to be constitutively internalized and not needed for the agonist-induced internalization, for example. And that is why I am calling them as the unique internalization mechanisms of mGlu receptor. But This is as a result of this work we just published in the paper in Cell Reports. But what we wanted to know is if mGlu receptor internalization it's a physiological process. And to that end, we proposed the use of glial cells from rats and white glial cells because it is widely described that glial cells have mGlu3 and mGlu5, which are both mGlu receptors that are constitutively and agonist-induced internalized, and also we have tools to investigate their internalization in native tissue. And how are we going to do that? Because before, I never say it, I think it's my mistake. We did all the previous study in hex cells. So the receptors are overexpressed, and we can manipulate them to express our enzymes. But in endogenously, we don't have these enzymes to tag them. So we propose to tag them with nanobodies, which are way smaller than regular antibodies. And the advantage of these nanobodies is that they are made by the team. We have nanobodies that they are neutral, so that means that they will not affect the function of the mGlu receptor. And also they have been already used to measure endogenous levels of mGlu receptors in the human brain, in the mouse brain, I'm sorry. So the idea was to label these nanobodies with the pH sense technology that has been released now from the enterprise Revity in 2025 in July. So this pH sense technology allows the labeling GPCRs with an antibody. So this antibody is tagged with the pH sense technology, which is a sensor that is sensitive to pH. So at pH 7, it's not emitting light. However, when the receptor is going in intracellular compartments, they are becoming acidic, and the fluorophore starts to emit light. So we don't have an antibody for these mGlu receptors, but we have a nanobody. So we label this nanobody with the pH sense technology. And we got this tool that is nanobody G3 pH sense, Europium. And we found out that indeed mGlu receptor is internalizing its agonist-induced internalized in glial cells. Which indicates that indeed mGlu receptor internalization seems to be happening in native tissue. And as a conclusion, mGlu receptor internalization is a fine-tuned biological process that could be modulated by endogenous ligands, drugs to ameliorate brain diseases. This is the photo that we took when I left in August, and my supervisors that they couldn't come. But I want to give you a little bit of an idea of why I'm here, because I've been studying GPCRs, but I am more and more interested about the part of neuroscience. I want to apply my GPCR knowledge to a more physiological context, and that context. And that's why I joined the team neurobiology lab the 1st of September to develop the project called Myelin Plus, in the frame of the Aristos program from CIVER. And the idea is to study myelin, because myelin it's a multilaminar sheet that it's wrapping axons to ameliorate conductivity and to give support to the axons. It is produced by oligodendrocytes, and oligodendrocytes are brain cells. So as brain cells, we thought, okay, there have to be a lot of GPCRs in oligodendrocytes, and indeed they are, and they can maybe modulate production of myelin. That's where Myelin Plus was born, to measure the impact of oligodendrocyte stimulation in myelin status and brain energy metabolism. And why brain energy metabolism? Because it's been not a long time ago that the team published this paper in Nature Metabolism, where they are measuring myelin content in the human brain, and they found that myelin can be used as fuel for the brain in the case of energy deprivation, like if there is not a lot of energy, like after running a marathon, myelin can be used as a source of energy, and then it will be recovered. And the idea, the hypothesis is that precise GPCR modulation in oligodendrocytes may modulate myelin architecture. And it was nice that even mGlu5 has been linked already to myelin shed growth. So it's nice for my perspectives, but I'm sorry because I have nothing to show you about this. I hope to come here in 2 years and be able to show you the advances of Myelin Plus. In the meantime, thank you very much for listening, and I am really happy to listen to your questions.
Okay, thank you very much, Marta. Very, very interesting and very new, unexpected, no, not agonist-induced internalization as well. That is a bit puzzling, but well, I had no time to go through your self-report paper. I just did a quick PubMed search and did not read it. But so let's see if there is anybody in the audience that would like to ask questions to Marta. So I can start asking a question myself. So so do you think this constitutive binding of beta-arrestin 2 to the mGlu3 receptor is going to be unique for the mGlu3, or do you think this, or have any hint of whether it can be working as well with other mGlu receptors?
Actually, I think it will not be unique, but breath measurements are limited because they depend. The breath transmission of energy depends on the orientation of the dipoles. So sometimes you can miss data if you are using this technique. So the idea is that a positive result, it's positive, but a negative result, it has to be taken more carefully. So the idea was to study, to try to do this experiment with HCRF, which is not depending on the dipoles. And we found that mGlu2 seems to be recruiting a little bit of beta-arrestin also in constitutive conditions. But from my point of view, it's right now, like we saw something, we need to move to native tissue, we need to move to neurons to see what is happening there, because that's what interested us, not so we have something to work with. Well, they have something to work with. It will be nice to see.
Yes, I think I see. When you, because of course I have no profound knowledge of your work and how this has been done technically, but so this this receptor, as you have studied it, is going on a constitutive internalization that will recycle to the cell surface again. So it's kind of is internalizing and going back, yes, constitutively depending on, I don't know what, because what is directing the constitutive internalization as well is kind of no? Intriguing.
Yeah, it's super intriguing. Actually, I've been 4 years and a half there, and during the 4 years and a half, I've struggled to measure recycling because it's really difficult. Because recycling, it's the receptor that has been already in the plasma membrane that is going inside and comes back. So this is really difficult to measure it precisely. We measure it after agonist-induced internalization, and indeed there is recycling of mGlu3 and mGlu5. But for constitutive internalization, we still don't know. For me, if you ask me what is happening with this constitutively internalized receptor, I would say it goes to the lysosome. Because
so it would be a way of kind of regulating, kind of taking the old one and putting the new one in the membrane,
exactly.
no kind of thing, not just because of functional regulation of that.
The cells are always alive, and if they are just producing proteins and proteins and proteins and they are not going to the lysosome, at some point they will explode. So there has to be some kind of system, and I would bet it's this one.
Of replacement of the old by new ones, kind of thing.
Mm-hmm.
Yes,
kind of thing.
Yeah. No, because I don't know, but still there must be something that is saying, okay, you are already, you've been here for long, then
yes.
Let's put a new one. No, kind
yes.
of thing. That is why there should be a signal for that as well. So for this constitutive,
there
maybe
has to be, but we don't know yet.
Mm-hmm.
I wish we knew.
Yeah. No. I mean, to me, because I like very much kind of trafficking, cell biology, and so really? It took my eye very much, and I enjoyed very much your talk and your results. I think is amazing. I don't know whether there is because I could do an additional question myself, but if please just don't be shy. Just participate making a question. The thing is that for training, we have to train as well just to talk with the speaker. That is an opportunity for all of us. And even though we can always phone her later or write to her and then speak, freely and more deeply into the details. But is anybody else wanting to ask or add any additional comment? Well, normally, I think we have to improve this participation this is going to be our goal for 2026. Just to get, at least the participation of some of the audience, but some people just don't feel like asking in a wider. Well, anyway, I was as well curious about, I suppose that your first studies, you say you were using HEC systems like expressing, knocking down the different components, but of course, it's true. I don't know about the expression of HEC or any other cells of this type of receptors, if they have any at all, or they are not really.
No, they express several GPCRs, but not mGlu receptors. They have no expression of mGlu receptors.
Yeah, so it's true that you need just to go to these nice systems that you are going to use now, and this will be really interesting to see what's going on as well there, and if you can observe this similar. I was going to ask you if you were using like physiological concentrations of glutamate just to induce the agonist-induced internalization, but I suppose that, of course, what is physiological, I mean, you could apply a physiological kind of a stimulation, but is not really a target cell for this. So I don't know what concentration are you using. If you tried different concentrations just to see the triggering of the internalization by the agonist exposure. I suppose as well the concentration or the stimulation of the different mGlus. It depends as well. They would be probably not the same, have the same sensitivity to the different concentrations of glutamate. So I don't know, how did you establish this to start with?
So it's a really tricky question because there is something that people don't know per se, or I don't know. I didn't know before coming there, and is that HEX cells produce glutamate. That means that if you are having a HEX cell living and you don't do nothing with it, it will produce a lot of glutamate that will activate your mGlu receptors because they are not used to have glutamate receptors. So they are just producing it, putting it in the extracellular media, and your receptor is always activated. You have to do a lot of tricks to be able to work in the absence of glutamate. This is not happening with astrocytes because they are ready to work, like they know how to do. But with X cells, that they are not brain cells, they are not used to that. So first, we had to optimize the conditions to really reach the minimum possible of glutamate. And secondly, we were starting always for a saturating dose of agonist, so a specific agonist or glutamate. And then we did dose response curves to know which was the potency of each compound for its receptor, yes. But we were starting always for a saturating dose to see if we could see something and then go try to decrease it. But it's also interesting because you said that, yes, indeed, every mGlu receptor has a different affinity for glutamate. For example, mGlu7, the potency of glutamate, it's stupid. You will never have this quantity physiologically. And it's still don't know why it is existing, because it's impossible that in a physiological context it will sense glutamate. But yeah, yes, every mGlu receptor is absolutely different, even for response to glutamate.